Bueno, buenas tardes. Good afternoon, everybody. PhD students, supervisors, and colleagues. Welcome to this second webinar of this series on research challenge in chemical engineering 2021 that our PhD program is organizing. First, I would like to thank the Fundación de la Universidad Complutense de Madrid for giving us the opportunity to organize this webinar. Of course, to all of you for your participation in this webinar, and very, very especially to our invited guest, Professor Jerry Kim, for his great generosity in sharing with his time and knowledge with all of us today. Well, as you know, the training of a doctoral student includes knowing in detail the research topics on which you as PhD students are working on. However, we believe that it is enriching to have a global vision of the different areas of research in chemical engineering. This being the main objective of this webinar series, that you have a better knowledge of the research challenge in the different areas of chemical engineering. Today we have the honor to have Professor Jerry Kim, that he will talk about the challenge and opportunity in the extraction of natural products. I don't want to spend more time on this introduction because I want to give way to the protagonists of these webinars. In first place, Professor Miguel Ladero, very briefly, is going to place today's conference in context with an important research area of chemical engineering, such as biotechnology engineering. Please, Miguel, the floor is yours.
Thank you very much, Carlos. And thank you to everybody for being here, and especially Professor King for sharing his wide knowledge on biotechnology and food technology with us. In these three webinars, we are talking about different fields where chemical engineering is being currently very active. One of them is biotechnology, up to the point that one of the branches of chemical technology nowadays is called biochemical technology. But obviously there are other fields like food technology engineering, where also biochemical engineering, biotechnology, and chemical engineering encounters. As you can see, biotechnology is a very wide and multidisciplinary field. Next slide, Carlos, if you can pass to the next slide. We can see different keywords that links chemical engineering, biochemical engineering, and biotechnology. And we can see that in these years, chemical engineering concepts are being applied to different fields, like the use of bioresources for creating biofuels and bioenergy, biomass-based chemicals and biomaterials, creating what is called the biorefinery. That is the refinery applied to bioresources, essentially. But biochemical engineering is mainly focused on the design and control of bioprocesses, and also on the application of biocatalysis and microbial biotechnology and green biochemistry to new processes. Also, chemical engineers are very active in cell and tissue engineering, genetic engineering, and also in environmental biotechnology. Obviously, all our knowledge are applied from bioproduct purification, separation processes, transport phenomena, and also obviously engineering of the chemical reaction. You can see that in the last 5 years, there are an enormous amount of entries on the web of science, including these topics I have been talking about. These topics are of such importance that now chemical engineering departments all over the world are changing sometimes their names so to include biochemical engineering or biotechnology in their names, and obviously in PhD programs that they create. On my side, this is all. I hope.
Okay, thank you, Miguel. Now, Professor Lourdes Calvo will introduce our invited guest today, Professor Jerry Kim. Lourdes, the floor is yours.
Good afternoon. I am delighted to introduce Dr. Jerry King and welcome him to our International PhD School program. I have known him for a long time. I really admire his work, which is why I have recommended him. I will provide a brief summary of his outstanding curriculum. Dr. King has over 57 years of experience in the field of chemistry, chemical engineering, and food technology. He has worked in government, industrial, academic, and consulting environments. Dr. King has published over 270 documents, including government reports and holds 3 patents. He is well renowned for his expertise and contributions to supercritical fluid technology in the fields of food, botanical, and natural product processing, as well as to analytical chemistry and material sciences. Dr. King is also a recognized expert in extraction engineering in chemistry, having made numerous presentations in this field. Emeritus from the University of Arkansas, he has been an R&D consultant since 2012 from his own company, Critical Fluid Symposium, with a focus on applying the above technologies in the cannabis and hemp industries, as well as other natural products. He serves on the editorial board of 5 international journals. Among many others, in 2020, he received the American Chemical Society Award for his research in the use of green technology related to environmental sustainability and consumer safety. Professor King, you have 45-50 minutes to summarize your enormous vision on the challenges and opportunities for natural product extraction. First of all, let me tell our attendants that they can ask questions to you via the chat or vote for those already asked in the section Preguntas. If they are in English, I will read them. If not, I will try to translate them to Professor King. By the way, Professor King knows quite a lot of Spanish, so it won't be very difficult even if we ask him in Spanish. Now your time, Professor King. We are ready.
Very good. I assume you can see my screen very well and hear me. This will be difficult to finish in 45 minutes, but I will do what I can. As you already realized from the introduction, I'm going to talk as part of your UCM webinar series in Advances in Chemical Engineering on the subject of extraction and natural products. And I have tried to focus this lecture on aspects of biotechnology and bioengineering, but basically it's a lecture particularly on chemical separations. So let's see, yeah. So challenges and opportunities. I put this out here because I felt like it was an opportunity to express concerns I have about what should be emphasized in the future. I taught chemical separations an advanced graduate course for a number of years at the University of Arkansas. One thing I became very aware of is that we needed to change a little bit our idea of our paradigm or how we do teach chemical engineering, because what you're really seeing today is an amalgamation or a merging of chemical with bio, and in particularly the case I'm going to talk about, food engineering. And I think also very important is for, based on my experience in research in academia and government, that the need for actual experimental data will always be there. And it's particularly pertinent to extraction processes as well as the pre and post-extraction unit processes, which some I'll mention. So even though we tend to be welded to theoretical methods and their prediction by software programs, and so forth. I really feel like the experimental data is important for the engineers to have some hands-on experience. Also, I learned very early that in chemical engineering curricula, most of these separation processes on the undergraduate levels were dominated by distillation, vapor-liquid equilibrium, and so forth. And that just isn't realistic in the future because particularly in the bio area, as you'll see, many other areas of separation technology are pertinent and need to be understood by a graduating chemical engineer. We also need to understand that there is a relationship in what we talked about in extractability. Oftentimes we focus a lot on theory and understanding the solubility of solutes that we're extracting in various solvents that I'll mention. But basically the matrix also plays a very key part in the extractability equation. So going on with this, I think that one of the trends I see today in a lot of the papers I referee and so forth is a base of like studying extraction on statistical based programs. And I think that this is a bit risky because it removes you from a sense of what on a molecular scale is going on, or a chemical scale. And just a dependence on essentially writing research papers and the results up in terms of statistics. I personally feel, as you'll see here a little bit, that really emphasizing solvent-solute structural correlations is, I think, highly desired. And as you'll see, I'll talk about extraction here. But also I'll talk about other separation processes, which are essential to gaining the final goal, some kind of purified solutes, purified chemicals. And these can include things like chromatography and membranes. Also, oftentimes in extraction, we can combine fractionation and also do reaction chemistry at the same time. And I'll cite a few examples of that. And of course, the scale up and critical assessment of the process is absolutely vital. I'll mention briefly some experiences as a Fulbright scholar at the University of Alberta, working with capstone engineering teams now for about 6 years, and show how this connects in with the principles of green engineering. So I don't want to spend a lot of time on this slide since it's kind of been referred to in the introductory comments, but these are all part of, I call it the biotechnology equation and bioengineering, taking these renewable resources, trying to do them in a green, environmentally responsible manner, looking at how we can use waste materials, particularly in the food and biomass industries. And also where needed to combine it with enzymes and environmentally friendly catalysis. So I think this collage of interactions here pretty well summarizes the critical areas I'm going to address. Now, there was no way when I looked at the magnitude of talking about extraction processes that I could cover the full gamut, so to speak. So this billet list here shows that this can be anywhere from what we conventionally experience first, liquid-liquid, liquid-solid, gas-liquid type extractions. And then the other areas get to be a little more vague to, I think, the undergraduate chemical engineer. They may not be exposed to mechanical expressions such as expellers and screw presses, let alone my favorite topic where I've done so much of my research career in sub and supercritical extraction. But there are also other techniques that are used in natural product extraction, such as pressurized liquid extraction. Some people in the analytical community refer to this as accelerated solvent extraction. And then there are assisted extractions such as ultrasonic, microwave, the use of enzymes in conjunction with an extraction medium, pulsed electric extraction. I will mention briefly an example of molecular imprint extraction and membrane assisted extraction. So forgive me if I haven't covered your favorite extraction topic here, but that just is impossible in the amount of time allotted. Now, in natural product extractions, this includes a variety of materials. And as you can see again from this list here, these are essentially items, particularly of human consumption, commercial interest, and can include spices, hops and coffee, particularly in the area of supercritical CO2 extraction, but also commodity and specialty plant oils, nutraceuticals that may contain omega-3 and 6 oils, such as the fish oils that most of us are familiar with, as well as antioxidants such as polyphenolics that are obtained from fruit matrices, or perhaps also, as I'll indicate, hydrolyzed biomass. And on the more topical side, cosmetic ingredients, perfumes, agents, and so forth are also fall under the area of natural products and so forth. And I certainly will mention in the latter part of my talk a lot of my current focus on aiding the medicinal cannabis industry, which is concerned with cannabinoid terpene and also hemp seed oil as a nutraceutical ingredient. So very quickly, I want to show you what some of these products look like. And this is biased towards supercritical extraction, particularly with CO2. But this is a collage of products. I was fortunate to visit this company in Taiwan a few years ago by Jobin Biomedical. All these are produced by extraction of natural materials such as sesame and ginkgo, and a few others that you can certainly read there. Moving on, this results in commercial products. This is a series of products that is marketed in North America by the New Chapter Company. These again are extracts done by supercritical CO2 on sea buckthorn, a forest type berry, particularly in the Nordic part of Europe. It's prominent and it's a source of omega fatty acids. It's important to realize too that these extracts do take quite a bit of effort and time, and not necessarily one type of extraction method is applicable. And here you see a propolis, which is derived from a bee honey source. And it is obtained both by alcoholic extraction, water extraction, and all these coupled again with supercritical CO2 extraction. So it can be multiple sequential extractions that yield the final product. This is an interesting product here. It happens to be a broccoli extract done again by high pressure CO2 extraction at around 600 bar. And it's interesting that from this you obtain a percentage of oil. You also, during the extraction process, sterilize, as far as microbial contamination, the Broccomax matrix or broccoli matrix. And in the process of doing the extraction under these kinds of conditions, one undesirable enzyme is deactivated, which causes off flavors. And another enzyme, marinase, is activated to essentially convert to bioactive compounds, as you see below. And I also put this in just as an example of extraction biotechnology at work in, again, a company in Taiwan. This is being used to extract bioactive ingredients from mushrooms. Again, in the upper left-hand corner, these are, in essence, done by CO2 extraction. So there's a lot of emphasis today on doing this in a green, environmentally consumer safe method, or using solvents that invest that. So I don't know whether you can see my pointer here, but a typical liquid solvent used in the natural products extraction area is ethanol. It has many advantages, particularly when it is coupled with water. It's called hydroethanolic extraction at atmospheric pressures. At higher boiling points and under modest pressure, it becomes a subcritical mixture. But as you can see on the left-hand side, you have agents. Most prominent one has been the CO2 extraction of a number of botanicals, natural products. But there are other supercritical fluids that can be applied, as you'll see on the next slide. So these can be combined in various ways to extract and also to conduct reactions simultaneously or sequentially. This is due to one of my Spanish colleagues, Fernando Montañez. It's simply looking at these fluid processing agents. And I emphasize the word fluid because it can cover liquid materials such as ethanol or propane or liquid dimethyl ether, which is what DME stands for, in combinations. And from this, you get a wide range of relative permittivity. This is essentially a change in the dielectric constant of the media you're doing the extraction with. So we have a lot of tools in our extraction kit to go after various molecules and compounds. This is from the engineering toolbox. It's simply an ethanol phase diagram. We're most familiar, of course, in using ethanol in this temperature range down here, where it's a liquid or a hot liquid. If we compress it in a bit with an external pressure force, it can be a gas or even hydraulic piston, we can essentially produce a subcritical fluid up to the critical point, but it will have a specific pressure required to keep it in the hot liquid state, as determined by the vapor liquid loci for ethanol. And then it is possible to do supercritical work with ethanol, but that normally is done not in the extraction field. One of the things that engineers may get a sampling of in the undergraduate level in thermodynamics, perhaps not, is the use of the reduced state concept or the law of corresponding states. And the inset diagram on the left side simply shows a plot of reduced pressure, reduced density, and of course the isotherms there that are reduced temperature. And as you'll see a minute in the next slide, particularly for water as an extraction media, a lot of different things can be done by looking at different reduced parameters. For instance, on the far right-hand side at the very high reduced densities, or basically liquid densities, we can do things like aqueous extractions, and one of my favorite areas, subcritical water extraction. On the other hand, if we increase the pressure and keep it in that temperature range, and again, you can see I've outlined this area where pressure and temperature can be varied, we have an ability to do reactions, particularly bioconversions of biomass for perhaps bioenergy or yielding carbochemicals. And finally, at extreme conditions, we can do gasification and oxidation of biomass materials. And this frequently is done under catalytic conditions and is called supercritical water oxidation or gasification. So what I'm going to talk about in the time I have is show you some examples of using subcritical water for the extraction of polyphenolics from waste grape pomace. We're also going to talk about using the same medium as an extraction reactive agent in the treatment of biomass using high pressure water. Also, I will briefly mention the extraction and concentration of omega-rich fatty acid oils, and you have some interesting technology in Spain in that regard. And finally, what has become very popular and certainly using various extraction solvents is the extraction of hemp and cannabis. And you've already seen some products, so I won't go any further on that. Food waste recovery is important. Certainly in my experiences in traveling throughout Spain and seeing your citrus waste and vineyard waste, etc., you have the potential of getting more economic value and environmentally treating these waste as additional, essentially, products. So biorenewable type materials can be, as shown in the top two inset pictures. You have your grapes. They're usually expressed somehow, and you have literally tons of this waste, still very colored, antioxidant-rich grape pomace. Likewise, in the field below is a field of switchgrass. And in this case, this is normally used with hot water treatment to produce essentially fuels such as ethanol. Solubility in subcritical water, and I've shown it here for a bioactive compound in a botanical called milk thistle, commonly is psilocybin. And unlike limited solubility up to about the boiling point of water here, by doing subcritical waters and doing them extractions and doing them rapidly, we can affect a much higher, as you can see, increase in the mole fraction solubility scale there. And this is a general thing that is true for all sorts of solutes as you increase the temperature and you keep the hot water pressurized. And this is some simulation work. We do simulation work despite my a little bit skepticism about computer modeling. And this was modeling some of the data we got on the milk thistle extraction, the bioactive component psilocybin B. And what you see here are two different vertical scales, the liquid phase concentration, that is what is in the extraction media or solvent. And here you have the solid phase concentration, and the arrows indicate which way they're pointing with respect to the graphs in this case. And the important thing to realize is that if you're doing extractions with liquid media at higher temperature, the speed of the extraction becomes critical in order to retain the molecular identity and efficacy of the milk thistle active component, psilocybin here. So all this shows is as you decrease the amount in the solid phase here, you have a point here about 30 minutes on this scale here to do the extraction efficiently without introducing something such as first order kinetics in degrading the psilocybin B. So I think I've said enough about the grape pomace connection. It also connects in with juices, and you'll see some work we have done with blueberry and cranberry juices here to produce valuable products by hot water extraction on the byproduct pomace. The reason why water interested us so much, and this was work that started at the USDA and I continued with a collaborator in food science at the University of Arkansas, is that traditionally this has been fed to animals or it's been composted in the earth. And in some cases, it can be fermented to yield ethanol. But the nutraceutical concentrate is much more economically viable. So the environmentally benign and sustainable process, which hot water extraction would be relative to ethanol, would be desired. Also in my country, and I'm sure similarly in Spain, the taxing agents, the IRS in our case, are wanting to know where that ethanol is going. You must keep a rigorous mass balance if you're a natural product extractor of the ethanol. So they know you're not diverting it to spirit production. So subcritical water has some great advantages. It also has some possible disadvantages. One of the things I'd like to just feature, we've done research in this and produced probably as many as 15 publications in the literature, is our kind of final act in this work, and that was combining extraction with a expeller. Now, you perhaps have seen or had some experience with an expeller, but here the grape pomace is entering here into the screw press, which is the helical press under pressure. And by controlling the temperature and heating the water here, we can effect an aqueous extract and exhaust the spent solids. And this is what the extract looks like, the water coming in. This is the essentially antioxidant-rich grape pomace extract with anthocyanin content, particularly very high. And this is the spent liquid after it exits the expeller, and this phase separation you see here is actually grape seed oil. So you can see we've done a good fractionation, relatively speaking, of this material. Now, it might occur to you at this stage that we've diluted the extract. In other words, in the natural products industry, nutraceutical industry, you want to concentrate. And so how would you concentrate this? Well, this is where other separation processes aside from extraction come in. So we did some work, and it's been published, on the concentration of hot water extracts, the polyphenolics in them with blueberry pomace, using different modes of nanofiltration. This happens to be a cross-flow system. I don't have time to go into the details, but whether we used this cross-flow system here shown in the laboratory, or we also used the dead end in the cross-flow mode obviously being desired because it's a continuous process. You can see for the nanofiltration membranes listed there, we got total polyphenol rejection, which is exactly what you want to concentrate it. So this, coupled with the hot water extraction, proved to be a viable process for winning the antioxidant-rich anthocyanin concentrates from waste of grape pomace and berry pomaces. We also wanted to optimize this, particularly for the extraction stage. So we looked at a number of different kinetic models, and I think it does right to look at a number of them and perhaps pick the one that not necessarily always fixes your data, but makes sense. And I'm going to show you a couple of examples of spleen fitting in what's called two-site kinetic models here. And this is the anthocyanin extracted from grape pomace on somewhat of a scaled up experiment here. You can see that it's almost exhaustively extracted after 10 minutes time. And you can see that by using the two-site model, we get a fair agreement between experiment and the modeling of this. This is not our work, but work done in Western Canada a few years ago on flax sheaves, a waste product of the flax agricultural industry. And they contain a very rich polyphenolic content. And again, we get a very good fit here using the two-component extraction model here between model and the experimental data. And you can see again the conditions below. The temperature is a bit higher than we use in grape pomace in this case. So in terms of doing these extractions, and generally a general chemical term used for these antioxidant-rich materials are flavolignans, particularly if they're derived from lignin type sources. And for the milk thistle, which I mentioned earlier, using hot water, we can use this spleen result here, which allows us to predict at what point to cut off that extraction. Because as I said previously, the problem is if you run the extraction too long and your solute or the compound you're trying to extract is left in the hot extraction media, you will degrade it. So this gives us a way of showing the spleen fitting being a two-component equation. We can then determine this. And likewise, we can do this again for another technique that we pioneered called hot-cold extraction on white Zinfandel pomace. But again, generally the spleen and methods work very well to minimize the extraction time. And I should say the hot water extractions are usually done in less than 15 minutes for most materials. Solvent feed ratios, of course, must be kept low. In this case, it was a SDF of 9. So we see a bigger role for this. And if you look at a grape biorefinery concept, and I'll show you on the next slide here, a portion of this particularly down here in the left-hand corner, involves the use of essentially a subcritical water extraction here. So from this, we can use these sub and supercritical fluids, depending on whether we're producing ethanol by fermentation methods here, or we can also do the subcritical water extraction on, say, the spent pomace. And this next thing shows the great juice press, like the one I was attempting to describe, or even the batch hot water continuous extraction. But essentially, it shows you the typical conditions and how the pomace can be treated after the wine or the juices are extracted here. And it can, as I say, be done on a batch basis, or it can also be done on a continuous screw press type basis. So if we extend this concept more into biorefinery, we have an ability to take these biomass fractions, de-oiled or unfractionated, and produce, depending on the amount of lipid material and proteins and carbohydrates, a number of things such as free fatty acids, depending on the degree and time of hydrolysis with this hot water extraction medium. We can reduce it to monomeric components such as amino acids or sugars such as glucose and fructose. Likewise, we can create mixtures here, which I don't have the time to go into, but can be used as a fermentic media to produce carbochemicals such as ethanol, or in the case of methanogenic bacteria, methane as gas. And I observed this a few years ago in an eco town in Japan and at Osaka Prefecture University. So the subcritical water processing of biomass, you would do your extraction generally in the range of 100 to 150 degrees centigrade for your botanical component. But it also, the biomass has the hemicellulose, lignin, and cellulose as the three major components. So these can be treated in much more severe extraction conditions, essentially to get a hydrolysate for fermentation purposes, such as the ethanol I mentioned. And if you look at the case of a very high lignin containing matrix, lignin's hypothetical structure shown here on the left, by hot water treatment, it can produce viable compounds such as vanillin, flavoring agent, and other antioxidants such as noted here in the third billet here. So from these, by chopping them up, you can get this value-added thing to these somewhat waste materials. The problem for the separation engineer is how to recover them from highly aqueous media. You can add high-pressure carbon dioxide to the subcritical water to accelerate this process and avoid the use of chemical catalysis like acid-base catalyst to use acidified supercritical water. And this carbonated water, as I like to refer to it as, hydrolysis with recycle here, and it's shown in this hypothetical schematic here. And again, having the lower sugars and the acids can be brought into a sugar fermentation scheme, in this case, to produce methane, which can be used again as a biofuel. And they do this in India and in Japan currently. And this happens to be a picture of the Osaka subcritical water hydrolysis plant located in their eco town. So hats off again, particularly to the Japanese with their dependence on foreign, particularly fuel sources, that they're developing these alternative routes. Quickly moving on to algae-derived extracts. They're of high interest because they appear in the nutraceutical and biomedical areas. They have some use in this case in bioenergy, although people I think have discovered over time that there's more value in obtaining essentially oils from algae that can be fed into the nutraceutical and natural products market. So these would include the well-known fish oils, seaweed, krill, and algae. There is particularly a nutraceutical product that is marketed worldwide that is obtained from a green lip mussel, and that is done with carbon dioxide extraction in the South Island of New Zealand. So marine waste products also are a rich source of renewable materials that can be treated by these various extraction methods that I'm attempting to describe. And they come out in various formats, all the way from the monomers, the free fatty acids, the triglycerides, but of particularly importance is creating ethyl esters, because ethyl esters can be extracted easier with non-polar solvents, be they liquid or something like CO2, than the more polar phospholipid-bound things, and also the first two components here. So most of the extraction from omega fatty acid formats do involve the extraction and reaction to form ethyl esters. And this results in these products here that proudly proclaim they have been extracted with, in this case, supercritical CO2, which provides a prophylactic effect, as we say in English, essentially the removal of oxygen, which these omega fatty acids are susceptible to oxidation, and the commensurate rancidity or off flavors that occur. And two companies in the European Union have contributed to this platform. I thought I would mention there is the Bexbach group in Germany called KD Pharma, and they involved the extraction with CO2 of the fish oil, and they first formed the ethyl esters in this case, and then they fractionated into enrichments of DHA and DPA and EPA fatty acids using supercritical fluid chromatography. So in both cases, you're using an ecologically desired extraction agent, CO2, in both of these unit processes. And in Melun, Spain, you happen to have one of these. This Spanish adventure started in the another company south of Barcelona in Tarragona, Spain, but now Salutex has the same process, very similar, running in both the SFE and the SFC to produce fish oil ethyl esters for the nutraceutical market from biorenewable fish oil feedstocks. Cannabis and hemp. Well, I'm running out of time here, but let me tell you my recent experiences. This industry sort of came to me because of their interest in CO2 extraction. And you look at cannabis and hemp with its 400, 500 compounds, and it is sort of a banquet for extraction and separation science. And I've simply listed all of the unit processes that food and chemical engineers could apply and do apply in this, in seeking out various products from cannabis and hemp feedstocks. The hemp plant is interesting. As I tried to show here, it contains seeds. It has one of the highest omega-3 and 6 contents in of all the plants available. The leaves, and particularly the flowers, are a source of cannabinoids such as cannabidiol and CBD that perhaps you've heard of, and is marketed, at least in North America, quite openly. And then the stalk and stems, again, represents lignin, cellulose, and biocellulosic compounds that can be treated, like with the hot water extraction process. And then you also have the roots, which are thought to have contained bioactive components, similar to what you might experience with ginseng roots. The complexity of cannabis, as I remarked, it has many, many compounds. And if you look at a part of this compositional pie here, the cannabinoids up here in the right upper corner here, and the associated viable and valuable terpene fraction only represent a portion. So part of the struggle for the separations, natural product oriented engineer, is to remove all these unwanted components such as chlorophyll and pigments, and particularly fats and waxes when processing by extraction of the cannabis matrix. This can be made simpler, and I can't help but emphasize this to engineers, by looking at the structure of the plant. And certainly the flower up here, which appears in the feminized cannabis plant or hemp plant, is a source of the very rich terpene and cannabinoid trichomes, as shown here in this little insect. The male flowers are killed off in the field, so they can't pollinate. And the result is the cannabis extracts that we obtain from these trichomes. This is a photomicrograph of the leaf here, and this is actually a one trichome. So one of the ingenious methods back in time that was developed is dry ice shearing of these trichomes by agitation and milling. And this results in a more stereospecific matrix that you can apply ethanol or CO2 extraction to. So this is just a bulleted list of the various cannabis extraction methods. They can involve cleaving the trichomes off and sieving them into a viable product. Mechanical pressing is done. Propane and butane, particularly 70-30 mixtures of 70% butane, 30% propane, or mixtures thereof, have been used. But as I think you recognize as engineers, this is a bit dangerous if not done properly, but it can be done with good equipment. CO2 extraction is very popular to produce concentrates for vaping and also other medicinal purposes. So we see CO2 used as a cleavage agent in the dry ice form. Liquid CO2 is used to sequentially extract the terpenes before using supercritical CO2 at higher pressures to isolate the cannabinoid materials. Ethanol and isopropanol, particularly isopropanol, are classic extraction solvents. Ethanol exhaustively extracts many, many components, and this creates a downstream purification product issue. So there's pros and cons of using these various methods. There have been some reported methods of using water extraction and also chlorofluorocarbon extraction, such as R134A. I don't want to spend a lot of time in this, but this is to illustrate, if we look at the cohesional energy density square root, a term known as the solubility parameter here, we can see that the cannabinoids represent a much higher solubility parameter than typical terpene components that are desired. And so the struggle in using different kinds of solvents, be they CO2 or propane, and you see some of the conditions listed here and over here, is that they are not selective enough in some cases to avoid overlap in compositions between the cannabinoids and the terpenes, which we want, and then also these contaminating materials here as waxes, triglyceride oils, and chlorophyll. So there are trade-offs here between selectivity and extraction and throughput. And you can see again that ethanol can be used very effectively as a solvent to precipitate the wax-like lipophilic components here, but it only overlaps to a certain extent with the cannabinoids, and it is a very poor solvent for terpenes. They're almost immiscible. This is a conventional cannabis and hemp extraction process from a company I did some consulting with. Essentially here you have an extraction. They prefer to use CO2, and then they do two different fractionations. They usually decarboxylate in this case to get a more neutral form, which is more compatible with the supercritical CO2 extraction. And then there are a series of other processes here involving precipitation of contaminants with ethanol, very cold ethanol, and also the application of molecular or short path distillation to increase the cannabinoid content. Normally, coming out in the crude extract, it's 35 to maybe 50%. You can, by evaporation, get it up to 75%, but by molecular distillation under vacuum, it can be increased to over 90%. And then if you desire a pure cannabinoid, such as cannabidiol, CBD, this can be done by chromatography, which is expensive to implement, but it can also be done by fractional precipitation in this case. One interesting thing I'd like to mention is the use of molecular imprinted polymers, or MIPS as they're called. These are essentially resin beads which are cast essentially with stereo cavities to reflect the dimensionality of the cannabinoids such as CBD. So when you do ethanol extraction, you get a considerable amount of contaminating material. And by running them through a column while you're doing the extraction, these molecular imprinted columns, molecular imprinted polymers, you can get a 70% or greater yield of, in this case, the target cannabinoid CBD. Finally, as I wind up my presentation here, I'd like to acknowledge some work we've done over the last 5, now 6 years almost, with capstone senior design team projects. These have been enormously gratifying to me to work with the senior level chemical engineers in doing work. And it just so happened, because of the desire in the University of Alberta to diversify from petroleum engineering kind of employment, that cannabis and hemp represented viable options to the practicing chemical engineer. So these again are the frontispiece of 2 different projects that I led. The first initial ones, they were fortunate enough to win actually a prize. And also now 3 or 4 years later, I've had a team that has worked on hemp seed oil extraction, looking at the benefits of ethanol versus CO2 extraction, particularly the effect of pressure on CO2 extraction. It makes no sense to do hemp seed oil extraction at, say, pressures of 2,000 to 5,000 PSI when you have infinite miscibility at 10,000. And so the hemp seed oil, because of its value on the nutraceutical market, and also the spent protein, which has a sellable market out there, doing it at high pressure makes a lot more sense. And this is typical example of a process flow diagram, which the senior chemical engineering teams did on the SFP of the hemp flower. What is interesting is the teams are divided into 6 members. One takes the environmental consequences, one does the cost analysis. Each has a specific assigned task. And again, they have a leader, and I interact with them frequently, say once every month, maybe twice a month as their projects become due. I think the need for physical chemical data is very great. And I put this in here at the end of my talk, mentioning all the work with cannabis, because there literally is very, very little data out of there. I was asked, for example, what is the temperature-dependent solubility cannabidiol, CBD, in ethanol? And it doesn't exist. Now, I know how to measure it, but I think one of the things that prevents academics, particularly in contributing to this field, are government regulations. Work that I collaborated with someone in New Zealand on, these chemicals like CBD have to be kept in a safe, they have to be accounted for. And so it isn't like doing research on some commodity product because of certain legal constraints. But all these kinds of data, I'd like to see actual physical data rather than just predictive data. But some of it does exist, like the pressure dependence, vapor pressure, CBD, and so forth. Finally, a couple of references. If my talk has resonated with you and created some interest, there is a recent book, I believe you can buy it as an e-book, called Natural Products Extraction, Principles and Applications. I feel this is a very outstanding book. It's part of the RCSC series in green chemistry by Rostago and Prado, who are at the Campinas campus in Brazil. And then your own work there in Computenza is very interesting, particularly in terms of understanding better how we can extract unsaturated fatty acids, which make up so oftentimes the beneficial compounds we want to extract from natural products that have omega-3, 6, and so forth content. And this is a work of Calvo and her team there. And finally, I do have a website. It's relatively up to date. You can access it. We certainly have a list of these 275 applications or publications that Professor Cobble mentioned. So I invite you to visit that. And finally, I want to express my appreciation to the professors and administrators at Computensa to allow me to address you. It's been an honor. I hope those of you who have listened in the Iberian Peninsula and perhaps even throughout the world have found my comments interesting. Again, thank you so much for sharing your time with me. And that kind of concludes my lecture. These are additional slides, but I think I've used my time.
Thank you very much for your nice talk. Now it's time for questions. If not, I will introduce some, a little bit of debate regarding green solvents.
Okay, there is one question, Lourdes.
Hold on, let me check.
Professor Jerry, Claudio Casella asked you, could one of these techniques you described today be used to remove microplastics from sewage sludge?
That's an interesting question because another part of my career, I dealt with plastics, particularly Los Alamos National Lab, and this was more in material science work. One of the interesting things we were looking at is by changing the density, say, of CO2, in this case, we could levitate, we could fractionate out polystyrenes and various polymers. The microparticulate plastics, in things like marine derived products, would be essentially a problem. In the case of the natural products, one of the advantages of using the CO2 is by leveraging its density and the temperature you do the extraction at, you would probably leave behind the plastics. If you wanted to remove them, there is solubility work and data that's been done on the solubility of polymers. Certainly the molecular weight is a negative factor in them being highly soluble. However, the use of destructive methods such as the hot water technique may prove to be viable in terms of destroying the plastics as you convert, say, a marine biomass into maybe a fuel. Those are just a few casual comments. It's an interesting question.
So Lourdes, I guess that you have a few questions also.
Yes, I wanted to introduce a little bit of debate. Water and CO2 are the greenest solvents possible. They are also available. And from your talk, we can deduce that they are the preferred solvents for you, also ethanol. But my question is, to use them, you have to apply high pressure. Is high pressure really green?
Well, the argument against the CO2 methodology is in the capital intensive expenditure for the equipment. Let me kind of rebuttal your question. There now exist at least 15 plants I can identify of some scale that are using pressures in the range of 800 to 1,000 bar. Astaxanthin, which is a very high valuable pigment having high antioxidant efficiency. I take a 4 milligram tablet every morning. It is done only at these high pressure plants. Uday essentially put in a plant about 5 to 6 years ago, maybe a little longer. That is a 2 by 250 liter plant, and it extracts the astaxanthin, which is a polar carotenoid from the algae. There are other types of materials such as phospholipids that are better extracted. Some of this comes from my consulting experience with the high pressure CO2. So there's essentially a trade-off is what is the final value of your product versus, the capitalization cost. But that has happened. The other comment I might make, is that the cannabis industry in particular has just exploded the application of CO2. Most of the equipment, there are over probably 30 vendors now. This does not include traditional vendors that you're familiar with in the food and maybe bioengineering area, but these are new companies, and they are limited usually to 350 bar, about 5,000 PSI, because all of them are made by welding technology. I would say that most of these new companies sell not just into the cannabis space, but also into other natural products extraction, because I think I forgot to say it in my preliminary remarks, but there's a trade-off between selectivity, which usually in the case of these pressurized fluids like CO2 is best in lower pressures, for example, several hundred bars of CO2 pressure. As you get into the higher pressures, you face the same problems as you do with ethanol, is you don't get a discriminated extract, you get contaminants along. And that is why combining essentially the CO2 extraction with fractionating columns, which I believe you're familiar with, or chromatography becomes attractive and viable, as I tried to illustrate in the case of the omega fish oil isolation.
Yes, the trade-off is between yield and selectivity, probably, right? That's the point. And we have another question here, also from Claudio Casella. He's asking if chlorofluorocarbon extraction, probably he means solvents, are safe to extract CBDs. Well, I will add something else related to this, is how nowadays are green solvent selected. What I mean is, which are the tools to select green solvents regarding this same question?
Okay, let me answer his question first.
Yes.
I mentioned the use of, I believe it's R134A. There is a company, they're actually based in the Balkans and Bulgaria. They market in the US and out of Arizona that uses R134A. There also are patents, again, this comes from consulting I've done, using other chlorofluorocarbons. Of course, these are environmentally somewhat undesirable, but also they're extraordinarily expensive and they must be recaptured. There's no question about that. The other another trade-off, if you want to call it that, or advantage to using them, is they can be pretty well used at ambient temperature in their subcritical state. So they don't require the high pressure vessels, and that cuts, in the cost of implementing an extraction skid or a plant in this case. There is a group in the San Francisco Bay Area region using chlorofluorocarbons to do cannabis extractions of cannabinoids and so forth. So again, you've got to weigh, by doing an economic analysis, the lowering capitalization cost. The cost of the fluorocarbons is much more, as is ethanol relative to CO2. Mm-hmm. You have to weigh those, and that's an important, I think, point. What parameters for greenness? That's a very interesting, insightful question. For example, I would give chlorofluorocarbon extraction pretty low marks, in terms of that, unless you can contain, the fluids and recirculate them. And that's very similar, for instance, to the use of propane butane mixtures in doing cannabis extractions. This has been practiced since the 1970s at least. But there is an inherent danger. So if you rate on a danger scale, what's more dangerous? Using propane butane mixtures versus CO2 at higher pressures. they both have considerations, but I think good mechanical engineering has alleviated some of those concerns. One of the questions over the decades I've always been asked is, does CO2 in doing extractions like the fish oil esters that I mentioned contribute significantly to the greenhouse effect? And the answer is no. In most cases, the CO2 is not recirculated. In the earlier days of CO2 extraction work, it was frequently saved, but it was frequently contaminated with water or other extracted solutes. So exhausting the CO2 is a relatively small penalty on the environment if it's connected with CO2 extraction processes. With ethanol, again, it's a chemical solvent, and whether you're using it for cannabis extraction or you're using it for the extraction, particularly in combination with water, for the antioxidants such as the grape pomace and similar type materials, hydroethanolic extraction is widely applied. The issues there again come down to the cost of the basic solvent and the need to reclaim it. You just can't go dumping the ethanol, into a stream in this case, which is one of the advantages of the CO2 process.
Mm-hmm. And another question that I would like to raise, because our students sometimes are not taught about this in the school, is how hard is to promote countercurrent contact between solids and fluids, mainly and especially at high pressures. You mentioned that in one of your talk, part of your talk, because you showed us on a slide where a screw was in countercurrent with the fluid. And I don't know if you can say a few words about the difficulty of moving solids in general and moving solids at high pressure for extraction, because natural products, biomass, etc., all are solids.
Right. Let's take the CO2 case. The earliest solution to moving the solids in and out were called lock hoppers. And essentially a lock hopper can operate under vacuum or ambient conditions, but essentially it's sort of a paddle wheel device that puts the hops, in the case here, or coffee beans into the extraction vessels, and then they are compressed. So it is, the entrance thing into the extraction vessel. Getting in our favorite use of the CO2, that has been existed for several years, both in Europe, particularly a group in the Netherlands comes to mind, as well as other contributors, the use of expeller screw presses under pressure to do CO2 extraction of solids, such as continuous soybean meal production. There were 3 plants set up in the middle part of the United States to do that. Cocoa nibs have also been done that way. So there have been developed engineering methods to do continuous solids addition. What we try to do in terms of really adjusting the space velocity in order to minimize the solvent to feed ratio, and also contact time. I mentioned hot water in the case early on in the talk, is that we want to have that residence time, to be minimal, anywhere between 10 or 30 minutes in the case of, hot water in this case. Otherwise, you're going to get, potential degradation, as I alluded to. So that's where space velocity and contact time becomes, something that has to be optimized in the engineering context. But there have been continuous devices such as lock hoppers, CO2 presses. I've actually experienced once in my long career the addition of dry ice in the hopper to like flaxseed or borage seeds. And as it goes in, it becomes compressed in the screw press. It goes from dry ice to supercritical CO2 under compression and heat. So all this takes engineering optimization, and that's where the chemical engineer I think, excels. May I ask a question back to you? In your classes at Complutense or in chemical engineering curricula in your various universities in Spain, do you have something comparable to senior design projects at the senior level that students execute, you know, as a team under faculty supervision?
Yes, they do have mechanical classes, mechanical engineering, not so much. They also have material science and design.
Mhm.
And obviously unit operations, obviously extraction, but we mainly study fluid extraction, not solid extraction, not solid fluid, I mean. And then at the end of the degree, they have to work together in teams in a whole chemical engineering project.
Okay. And they have to write a report.
Yes.
And use
a technical report and
Okay.
present it in front of a panel. Yes. Yes.
That's very
And
good.
probably, I don't know if we have more time, but the last question that I wanted to ask you is, you mentioned that extraction, and also Professor Ladero mentioned that extraction is a primary isolation step in a whole process where it is important downstream operations, for example, membranes or distillation to concentrate extracts, and also it is important the preparation of the raw material, especially in natural product extraction. Could you tell us a little bit about this, because you didn't mention in your talk the preparation, the pre-treatment of the raw materials, how important it is for extraction?
Yeah, I sort of skipped over. I can't easily bring my talk back up. Maybe I can, but let's see if I can share the screen again. Maybe not. Yeah. I think I have something here that may work. There. Let me go back to the schematic here. Excuse me. Yeah, like here. Here you see the dry bud. There's actually a better schematic of this. I don't know whether I have it included here, but generally this, like I tried to emphasize, you've got to look at the structure of the material. First of all, you know, I have had people in the cannabis industry that just grind up the entire biomass from the cannabis or hemp plant, and that's not necessarily the way to do it. What you want to do is to segregate the best you can before you even begin to do an extraction, which results in a concentration of a bioactive component. Believe it or not, this can be done simply by pulling the buds out. They're called bud tenders, and this is how they get granular marijuana for your cigarettes. But in most of the cases, milling is critical to get a certain size, and not necessarily the finest milling to get the finest particle size, which can cause back pressure and channeling in the extraction vessel, but also to get an optimal particle size, in this case, that is commensurate with getting an efficient extraction. Drying will be very important in the case of, say, a hydrocarbon or a CO2 extraction. In the case of CO2, if you're dealing with a very hydrophilic containing matrix, the water will sterically inhibit the contact between the extraction molecules, the CO2 in this case, or virtually create an immiscibility case if you were doing hydrocarbon extraction. In the case of something like an aqueous extraction, one of the advantages in the expeller process I alluded to was that you can don't have to dry the material. Mm-hmm. So that's a great advantage. In fact, you get a better recovery and selectivity by using the wet material. However, the caveat is you must do it quickly, because if your material sits around too long, it will ferment into ethanol, and that would not be desired in the case of trying to get your nutraceutical component. So I think things such as drying, sifting, comminution, or essentially grinding these become very important. But before you get taken away too much on that, you have to consider, is there some way you can segregate a certain area of the plant that contains the target you want? Years ago, we did a desert botanical plant called the desert smoke tree. It turned out this perfume type ingredient, very similar to the thing from India, sandalwood, was actually in the calyx. It was actually in the outside of the buds of the plant. So grinding it up simply contaminated and made extraction and purification more difficult. If you just took the desert smoke tree seeds without grinding them, you got out the desired semi-volatile perfume ingredient. So I don't know, that's a lot of verbiage on my part, but you're certainly correct. It's very important to sort of respect the morphology of the plant material you're dealing with in this case. And before you do too much sample prep, as you might say in a laboratory work, that you consider the, the pros and cons of advantages of that. I hope maybe I've made myself,
Yes. I would like to point out that drying and milling is very expensive.
Mhm.
This is why these two previous operations of natural extraction should be always optimized, as you mentioned.
One of the things that interested me, again, just in terms of time, let me bring this back up here, if I can get it. These are slides that I didn't use. I don't think you can see them now. I guess we've stopped the slide sharing, but you have these cork plants in both Portugal and Spain, in which the treatment is done on a batch basis to essentially sterilize and also to kill an enzyme that produces an off-flavor component, namely an anisole-based chemical compound that creates off flavors in the wine industry. And this is an interesting example. And again, both particularly Spain and Portugal have led the world in this type of thing. But again, my understanding is that before they're pressed into the wine bottle corks, in this case, it's done on comminuted cork, that's been very finely ground to affect the treatment. And again, this relates back to considerations of diffusion and mass transfer, which usually in the supercritical region, whether it's CO2, are much improved over liquid CO2 or liquefied solvents.
That's right. Yeah, you also mentioned in this case and in another example, the importance when treating biomass in general or wastes, the importance of the microbial count, the microbial contamination previous to the extraction, during storage of the raw material and also of the product. And this is also something very important that has to be considered previously to start an extraction.
I couldn't agree more. Yeah. Another thing I didn't include, again, you can't see my screen, but I have a slide here where an extraction vessel was filled with 50% hemp seed, which has a high omega oil. And the other 50% by volume was with the flower buds from the hemp plant that contained the cannabinoid. And out of this came a very light colored sort of waxy gelatinous mixture, which had the benefits of both the omega fatty acids coupled along with the cannabinoid and terpene content. And the message I guess I want to say here is, I think in terms of chemical engineering phase equilibria, we need a better understanding on the effect, not just of co-solvents like ethanol when they're combined with CO2, but also the effect of the extracted material. In other words, these ternary or three component systems, because essentially they can influence ultimately the selectivity and the yield that's going to come from the extraction in this case. I don't know whether I've made myself clear on that.
Mhm.
I just think like Professor Timelli in Canada, she has studied a lot of oil extractions, but she's particularly interested not just in the interaction of CO2, but how the components interact along with the CO2 and the targeted compounds. The co-extractives, I guess is the word I'm looking for, can have an advantageous advantage in some cases to increasing your yields if you can allow them in your final product. And in the case of a hemp seed oil being co-extracted along with the hemp flower, this produces a desirable product.
Yeah, what you mean is that sometimes from biomass and from other natural products, other co-products that can be extracted together with the main product can help the extraction, and some other times can be negative for the extraction. So probably what is needed is to understand the phase liquid, the phase equilibrium, and how these other co-extracts will perform during the extraction. This is what you meant?
Exactly. Yep.
Yeah. Natural extraction is very complex. It's very complex because of all these reasons.
Yeah.
I don't know, but probably it's time to finish up, Carlos. How are we doing?
Yes, I think we need to finish very soon. May I comment something? Well, this topic is outside my research expertise, no? So for me, it has been very useful because while I get an overview about what is going on, I impressed about the number of high pressure plants that they are already in operation. You mentioned a very high number, no? It's always to obtain high value products.
Almost, I would say, yes, there's a synchrony between that because of the capitalization cost.
Yes.
The plant I cited, I wish I could go back to that slide. Again, it was one of the slides I eliminated, but these are multi-million-dollar plants. But again, the product they're producing, astaxanthin, when it first came out as a nutraceutical supplement, was $10,000 a kilo.
Yeah, of course.
That gives you an idea of it.
And they are also low capacity production, low production capacity, or not?
They're fairly high capacity. I mentioned the one that I'm familiar with, the scale that Uday in Hagen, Germany produced for the company Cyanotech.
Yes.
They have a 2 by 250 liter capacity. Another company that offers some degree of scale up is in Bavaria in Germany. I think it's called NATE CO2 or natural CO2. They have 1,000 bar capability. There also are plants operating in my country, in the area of Orlando, Florida. It's an Indian-owned company, but it started as a US-owned company. They have capacities to do 800 bar extractions, and they offer a variety of products. The main thing I would comment on is that once CO2 finds a niche and people are aware of its, safety value to them and also environmental issues, it captures, like all of hops, extraction. Every hop essence that is produced in the world is done by CO2 extraction. There's no question. All astaxanthin is pretty well done that. Another comment I made, and saw palmetto berry is entirely done by CO2 extraction. And in my long career in this field, it's very impressive to see the array of products coming out now. In the late 1980s, in the area of cosmetics and perfumes, there was little interest in CO2 extracts. They were more expensive, among other things. But over time, that has changed. Even if the CO2 extract or the perfume extract was superior, the industry would not accept them. That has changed quite a bit now. And I didn't mention it, but in the area of cannabis extraction, the cannabinol materials and also the what are called the minor cannabinoids, they find value particularly in topical applications for pain relief and certain other kinds of diseases.
Okay, a very last question from Albertina Cabañas. How important is temperature when extracting natural compounds to keep active components?
It's very critical to have an appreciation of it. I showed how that the speed of extraction was good with doing like hot water hydroalcoholic extraction. You really have to be sort of aware of measuring two things: the loss of a material due to operating at too high a temperature, and also the adjustment of the space velocity through. You want the lowest, of course, solvent to feed ratio you can get by with, and that will give you the best concentrate, you know, in most cases. But temperature is very, very critical. Again, each natural product matrix, you know, has to be kind of experimentally that determined, you know. For instance, the extraction of terpenes from these cannabis and hemp matrices is usually done a little below the critical temperature of CO2. Typically, it might be 25 to 30 degrees centigrade, whereas the temperature is increased substantially for the cannabinoids. That's because with CO2 extraction, the actual extraction efficacy is a product not just of the solvent density, which is linked to the pressure and temperature, but also very much to the vapor pressure of the compound too. That's why we need vapor pressures on cannabinoids. And by the way, there is vapor pressure data that's been determined by the National Institute of Science and Technology, NIST, on THC and on CBD. That does exist. What would help is to have essentially vapor pressures under vacuum, and some of that data has. I actually have a talk that I've given on what is available in natural in terms of physical property data and cannabinoids. There is a ton of information on terpenes, but it comes from other fields. Particularly investigators in Portugal have done a lot of work on citrus oil components, you know, like limonene and other terpene or sesquiterpene components that also are in the hemp and cannabis matrix, the same compound. So that can be a source of valuable information on physical property data.
Mm-hmm.
Okay, thank you. I think it's time to close. Well, Professor King, has been an honor and a privilege to have you during this webinar with us as a guest.
Yeah.
I greatly appreciate it that you have shared your time and your knowledge. I know that you have many commitments and you give many conferences and a completely full agenda. So thank you very much.
Well, thank you.
I also thank Miguel and Lourdes for moderate this section. Well, to the staff of the Fundación Universidad Complutense for making this webinar possible. Last but not least, of course, to all of you for your participation. And finally, I would like to remind you that our next webinar will be on December 14 at 4:30 p.m. And our guest speaker will be Professor Ignacio Grossman, who will talk about advanced optimization for process system engineering. Thank you to everybody. Enjoy the evening and see you soon. Bye.
Thank you. I hope to see you in Madrid shortly.
You are welcome, of course.
Bye. Thank you. Gracias a todos.